Hi, this is Dr. Roshin CN, Assistant Professor, Department of Oral Pathology and Microbiology, Mahi Institute of Dental Science and Hospital. I'll be discussing about the fungal infections today. So, as you all know, mycology is the study of fungal infection, which has gained remarkable impetus in the past few decades, owing at least in part to the fact that fungal diseases are far more common than that was previously suspected. Thus, the importance of the oral fungal infections has increased during the last decades. The fungi are also components of the normal oral microbial flora. Both yeast and the filaments type can also cause infection of the different part of the oral cavity, which includes palate, buccal mucosa, tongue, and as well as gingiva. So it is important to study the fungal infections because dental practitioners can come across these fungal infections in their routine practice, and if not treated, it can lead to fatal complications. So, as I have mentioned before, the study of fungi is called as mycology. The term mycology is derived from the Greek word mycos, which means mushroom. This is the basic characteristics of a fungi. Fungi are eukaryotes. They are multicellular or unicellular organism, heterotrophic and consume organic matter. They lack chlorophyll. The cell wall of the fungi contain chitin, and these are non-motile organisms. And eukaryotes are organisms whose cells contain a nucleus and other membrane-bound organelles. So, fungi also have nucleus as well as the other or membrane-bound organelles. This is the basic structure of the fungal cell wall is very important. So fungal cell walls are rigid and contain complex polysaccharide called as chitin, and it also contain glucan. The chitin gives or adds structural strength to the fungal cell walls. So this cell wall composed of a polysaccharide based three-dimensional network. So it's considered for a long time as an inert circle. But the cell wall is now seen as a dynamic structure that is continuously changing as a result of the modification of culture conditions and environmental stresses. So, although the cell wall composition varies among fungal species, chemogenomic comparative analysis have led to the better understanding of the genes and mechanism involved in the construction of the common central core composed of a branched beta-1,3 glucan chitin as well as beta-1,6 glucan chitin complex. So, because of its essential biological role, unique biochemistry and structural organization, and the absence in the mammalian cells of most of its constitutive components, the cell wall is an attractive target for the development of new fungal agents. So, genomic as well as drug studies have shown that the death of the fungus can result from the inhibition of cell wall polysaccharide synthesis. To date, only beta-1,3 glucan synthase inhibitors have been launched clinically, and many more targets remain to be explored. So this is the basic structure of a fungi. It has got a fruiting body and a below-ground structure called as mycelium. So most fungi have a filament structure. They can be single-cell or multicellular organism. Fungi consist of long thread-like bodies, which is called as hyphae. Uh, so these hyphae form a mesh-like structures called as mycelium. You can see here the mycelium. The cell wall is made up of protoplasm, which is further comprised of other cell parts like cell membrane, cytoplasm, cell organelles, and even nucleus. So the nucleus is dense, clear with chromatin threads and surrounded by a nuclear membrane. Again in detail. So hyphae, they are thin filaments making up fungus, some called as rhizoids, which helps to anchor the fungus. You can see here hyphae and mycelium, where the mass of tangled filaments is hyphae. So most of the mycelium are seen in the soil or embedded in the host tissue. You can see the floating body here, and they are the reproductive structure of the fungi, and there are spores which is thick-walled located at the tip of the hyphae. This is mainly for the reproduction purpose. Moving to the classification of fungi. So based on the type of fungus, hyphae can be one large multinucleated cells when they are called as non-separate hyphae, or it can have a dividers between the individual cells when they are called as separate hyphae. So this is a coinocytic hyphae or the non-separate hyphae, and here you can see the separate hyphae. So first I'll discuss about the septate hyphae. So septate hyphae have dividers between the cells, which is called as septa, singular septum, and the septae have openings called pores between the cells to allow the flow of cytoplasm and nutrients throughout uh, their mycelium. So although the septae separate the cells in some hyphae, the cellular components including the nucleus, they can fit through the pores. When new cell bud at the apex of the hyphae, a septum does not form immediately. So as a new cell matures, the cell wall grows down into the cytoplasm, which forms the septum. So members of these classes are like basidiomycetes and ascomycetes form septate hyphae. Next is non-separate hyphae. Non-separate hyphae is also known as aseptate or coinocytic hyphae, which form one long cell wall with many nuclei. They are more primitive form of hyphae. Species with septate hyphae diverge from a common ancestor with coinocytic hyphae. So most fungi with coinocytic hyphae. Uh, so while they do not form septum between the nuclei, they do form a septum at branch points that connect one filament to another, which preventing the entire network from being compromised if one hyphae is injured. So the non-separate hyphae is also known as coinocytic hyphae as well as aseptate hyphae. So based on the morphology, we can classify uh, fungi into yeast-like fungi, molds, and dimorphic fungi. First, I'll discuss about the yeast. So yeasts are unicellular, and it reproduces by budding, which is usually seen as spherical or in oval forms, which forms the bacteria-like colonies. So the example is Cryptococcus neoformans. Next is yeast-like fungi. So they are unicellular, which reproduce by budding and fission. Like yeast, it's also seen as spherical or oval form with filamentous or hyphae, like that's called as pseudohyphae. So it is seen as a spherical or oval forms with filamentous or hyphae, like it's also known as pseudohyphae. And elongated cells join end to end with constriction. So, but fail to detach and become elongated. One of the examples of yeast-like fungi, or common example for yeast-like fungi, is Candida albicans. This next is very important. There is mold or filaments fungi. They form true mycelia and reproduce by the formation of different types of spores. The best example is Aspergillus. The hyphae, they have thick-walled tube-like extension with fungal cytoplasm and organelles, usually 2 to 10 micrometer in diameter. And mycelia, they are tangled, intervened hyphae. So next is the last one is the dimorphic fungi. They occur in two forms. One is in mold form, other one is in yeast form. So most fungi which causes systemic infection belong to the classification of dimorphic fungi. So you can see here there is a switch between the mold and yeast. So the ability of pathogenic fungi to switch between a multicellular hyphal and unicellular yeast growth form is a tightly regulated process known as dimorphic switching. So a dimorphic switching equates the fungus to sense and respond to the host environment and is essential for the pathogenicity. These are the examples of fungi, which are dimorphic fungi. So you can see here at about 37 degrees Celsius, it is appears as a yeast, and at 25 degrees Celsius, it appears as a filamentous. So based on the temperature, the organism will switch from one form to another. That's why it's called as dimorphic fungi. Next is oral microbial flora. So the presence of nutrients, epithelial debris, and secretion makes the mouth a favorable habitat for a great variety of bacteria. Oral bacteria include streptococci, lactobacilli, streptococci, and corynebacteria with a great number of anaerobes, especially bacteria are seen in the oral cavity as a normal oral microbial flora. So the mouth presents a succession of different ecological situations with age, and this corresponds with changes in the composition of the normal flora. So 50% of them shows the presence of Candida species. There are so many risk factors or predisposing factors associated with fungal infection. They are mainly the drug therapy. Certain drugs can even cause the fungal infection, and blood dyscrasia is malignancy, a dietary factor, that is nutritional factors, and certain endocrine disorders, even the immunological disorders and salivary changes can also predispose to the formation of fungal infections. Cigarette smoking also predisposing the formation of fungal infection in the oral cavity, and usage of inhalational steroids. Extreme of age, that means it is seen in kids as well as in the older aged individuals. So danger wearing also an important factor for occurrence of the infection, mainly Candida albicans, which will cause the danger stomatitis in danger wearing patients. So that is about introduction and characteristics of fungi. Thank you.